Good morning, everyone. We're going to wait a few more minutes to see if anyone else is going to join us for the May Coastal Beach Water Quality Workgroup meeting.
Good morning.
Morning, Matt. Hey. Well, thank you everyone for joining. I'm sure a few more people might pop in over the next few minutes, but let's go ahead and get started. So this is the State Water Board's May Coastal Beach Water Quality Workgroup meeting. My name is Kat Walsh. I supervise the Ocean Standards Unit in the Division of Water Quality. And I have a few folks from my team here. I have Michelle Robbins, who's an environmental scientist on our team. And I have Amanda Blackwell, who is our newest environmental scientist. She is going to be taking over as the lead of the Coastal Beach Water Quality monitoring program at the State Water Board. Amanda, do you want to take a few seconds to introduce yourself to everyone?
Yeah, sure. So hi, everyone. I'm really excited to be here and be a part of this program. My education is in international environmental policy. And then for the last few years, I worked at Cauliphage, supporting their business services office. And then I went on to do contracts at Department of Water Resources. So I'm hoping to, lend those skill sets here and help you guys out, in this program. So really happy to be here. Thanks.
Thanks, Amanda.
Recording in progress.
And so you all probably heard we are now recording the meeting. And we will move on to roll call for the meeting. So we have folks from DWQ just introduce themselves. Is there anyone joining us from the Office of Information Management and Analysis of the State Water Board?
Nick Martorano's here.
Nick.
Morning.
Anyone else from the State Water Board or the Regional Water Quality Control Boards? Okay, hearing none. We'll go around the local agencies next. Anyone joining us from East Bay Parks Department?
Hi, this is Hal McLean. I'm from East Bay Regional Parks, and we do water quality at Alameda and Contra Costa County, Crown Beach, and Keller Beach, respectively. Thanks.
Thanks, Hal. Has anyone joined us from Humboldt County? Hearing none. Has anyone joined us from the County of Long Beach?
Hi, this is Keith Allen, Supervisor Environmental Health with City of Long Beach.
Thank you. Anyone else joining us from Long Beach?
I think I'm it.
Okay. Has anyone joined us from Los Angeles County? None. How about from Marin County?
Oh, it's not my name. This is Kylie He. I'm the
Hi, this is Keith Allen, Supervisor of Environmental Health with City of Long Beach.
Great. How about anyone from Mendocino County? Anyone from Monterey County? Is anyone joining us from Orange County?
Hi, my name is Lauren Robinson. I'm the program manager. I also see that we have a bunch of people, John Pang, Joe Guzman, Sham Almashad, who else? We have Grant Sharp. Did I miss anyone? Anyone else on here from Orange County?
Great, thank you.
Gina probably going to join us a little later.
Okay.
Thanks for joining us. Has anyone joined us from San Diego County? None. How about from the city or county of San Francisco?
Yes, good morning. Diana Downey here from the city. In cooperation with the San Francisco Department of Public Health, we monitor all the 21 sites now around the perimeter of San Francisco. And my cohort is listening, but he's not in the Zoom meeting today.
Thank you. Anyone joining us from San Luis Obispo County?
Good morning. I'm Leslie Terry with San Luis Obispo County. I'm a supervising environmental health specialist here with program responsibility for our ocean water monitoring program.
Thank you for joining us. Anyone here from San Mateo County? Hearing none. How about from Santa Barbara County?
Hi, good morning. This is Marissa from Santa Barbara County.
Good morning. Anyone joining us from Santa Cruz County? How about Sonoma County? How about from Ventura County? Great. Anyone joining us from the EPA or any other agency that I may have missed? Great. And we have a couple of other folks with us here today who will be presenting. You're welcome to introduce yourself now. I'll also introduce you later if you want.
Sure, yeah. You can introduce me before I present, but my name is Matthew Verbelia from San Diego State.
Thank you, Matt. Great. Oh,
I was gonna introduce myself too. I'm one of Matthew's students, and I'm also an intern at the San Diego Regional Waterboard. And my name is Eric. Hi.
Good morning.
Hi, good morning. I would like to introduce myself. My name is Aarti Kundu. I'm here representing Environmental Health Services, Marin County. I'm a project manager. We monitor 31 beaches in Marin County. Glad to be here.
Thanks, Aarti. All right. Well, just a reminder for everyone, if you are listening but not participating in the Zoom, but you would like to, you may email the Waterboard's beach program email at dwq-beach-monitoring@waterboards.ca.gov and request a Zoom meeting link, and one will be provided to you. A portion of our meeting today will be a closed session with the local agencies. So if you would like to participate in that discussion, I encourage you to request a Zoom meeting link if you haven't already done so. And then also a reminder that we do have this beach program email set up. It's going to be really particularly helpful for everyone to continue to send their inquiries, their invoices, things like that related to the beach monitoring program to this email, as there are multiple people who will be responding and processing your invoices. Amanda will be the lead, but if she's ever out or she needs assistance, Michelle or I will be helping as well. So it's really helpful for us if you email the DWQ-beach-monitoring at waterboards.ca.gov email account. Couple other reminders to share with everyone. Our current grant agreements are ending on June 30th of this year, and we're currently working on new grant agreements. If there are any documents you need to provide or things that you need to confirm for our team over here, it'd be best if you could do so as soon as possible. I also want to share that the State Water Board is in a joint effort with CASQA hosting a bacteria summit this September. It will be September 14th through 16th. And the intended audience is water board staff, the regulating community, the environmental community, academics, and the general public. And the concept is, it's recognizing that there's new information on methods, epidemiology, risk, and that there are, a couple of different approaches that can be taken to update our standards or address regulatory permitting issues, things like that. So the goal of the summit is to identify priority technical and regulatory actions needed to safely recreate and safely eat shellfish, and to process and implement those actions. So that bacteria summit again will be hosted from September 14th through 16th, and more information will be released over the summer. And now I'll open it up to the local agencies and other folks on the Zoom call for any updates or information they would like to share with the group. Does anyone have anything going on with their beach monitoring efforts that they want to share? Any challenges that they're experiencing that they want to discuss?
Hi, this is Hal McLean, East Bay Parks. We've had a surprising number of exceedances this year, and I think some of it has to do with I don't know, turbidity and windy kind of weather related, because we don't necessarily see a lot of people on the beaches when we're sampling. But yeah, I'm assuming my grade's going to go down with Heal the Bay, because we have 17 resamples out of 86 total samples. So that's kind of interesting. And we've been getting lots of, we do MFC and MEI filtration methods for fecal and enro. And we've had lots of congruent growth results, which is maybe along the same lines. But that's kind of interesting. We also do freshwater testing, and the cyanotoxins have caused advisories that have closed 2 of our freshwater lakes already. So, and this kind of happened last year too. But we did have some closures in November and December and February, which, you know, it's always interesting. I guess that's California for you. And we started doing some DNA source tracking on one of our other freshwater beaches because of high E. coli. And I'm going to knock on wood, but we haven't been getting elevated E. coli since we've been really trying to take samples. Anyways, yeah, if anybody wants to talk about that, let's talk in the chat or something. Thanks.
Thanks, Hal.
This is Fernaz Farhang with the County of San Diego Beach and Bay. The most recent update that we have is that we started implementing our Ddpcr method, which is exciting, last Thursday on the 5th, and that has started being done on a daily basis.
Congratulations, Fernaz.
Hi, this is John with OC Public Works, Orange County. Just a couple of things going on. So we have a bacteria TMDL, fecal coliform TMDL for the Newport Bay since late 1990s. So we have been implementing it for 2 decades now. After the deadline of the TMDL passed, we have a time schedule order to do a number of things. One of the things that I'm leading, the project I'm leading, is the source investigation focused on the human sources. So we have a really great team of consultants and other colleagues from healthcare agency led by Lauren and Joe Guzman. So basically, we try to look at potential sources of human, like homeless encampment, leaky sewage, private laterals, or the septic tanks. So we started the effort in February, and it's been going on for 3 months now. We did find some sources that are things like homeless encampment and private laterals. So these are the things where, turn out to be pretty difficult to cure, at least in the long term, right? Because, some issues come back even if you fix it. So we will wrap up the study in by the end of this year, and hopefully by then we can report back the findings. That's one. We also, interestingly, for our fecal coliform TMDL, we have a shellfish component as well. So part of the efforts of the watershed stakeholders is to see how the shellfish beneficial uses are protected, even though there are lingering issues with the beneficial uses. So we, with the help by SQUIRP, we've done phase 1 of the shellfish study where we didn't actually see a linkage between the fecal indicator bacteria in the water and the tissue of the shellfish. So that's the phase 1 is for dry weather. So for wet weather, we are planning to kick it off this winter to look at winter season correlation between the fecal indicator bacteria and pathogens and other indicators in shellfish. So it's going to be a pretty major effort, probably will last a couple years. So hopefully by then we have some exciting results to share with the group as well.
Thank you for that.
Hi, Hal. There was a question in the chat for you. Are you uploading the resample results to Beachwatch?
Yes, we are.
Great, thank you.
Any other updates, items you want to share with the group, questions that you want to raise, anything like that? All right. Well then, I think we are ahead of schedule, but we can move on to
Hey, Kat, can I
Yeah.
just make a quick announcement?
Chris. Yeah.
So I think everybody already knows, but Heal the Bay is planning on releasing the annual beach report card. The tentative date we have right now is June 15th. I believe I've spoken to all of you already, but just wanted to kind of reiterate that right now. So June 15th, it might be later than that though.
Thanks, Luke. Okay, one last call for questions, updates, information sharing.
Hi, this is Mario Calson. I'm from Humboldt County.
Hi, Mario.
And if this is a good time to free share, I think I missed the request for reports out from counties or regions. But I wanted to share a little bit about our surface water quality sampling program and our beach monitoring program. They're closely tied together with our LAMP, our local area management program for siting and regulating on-site wastewater treatment systems. And the beach monitoring happens at some beaches which are impaired for bacterial loads. So we monitor those in the summer months and get results all over the place, and we're not happy with the results, and we're not happy. Yeah, we need to have better understanding of the mixing zone and the impairment and things that are affecting the mixing zone. And so what we're doing is sampling the surface water tributaries that drain to those beaches. And what we're finding, we're sampling for fecal coliforms, total coliforms, fecal and enterococcus, and we're also doing some microbial source tracking. And in the winter months, we have sort of two regimes for the surface water sampling of the tributaries. One is after the first rain in the fall and a first significant event. So 1/2 inch in 24 hours would be a significant event. And again, we sample at the latter end of the winter months. This year, it wasn't very wet because we had a long dry period of January and February, but then it started to rain again. And we did get a sampling event of, again, 1/2 inch or more in 24 hours. And so we sampled, I think it was late March. And we are seeing high elevations of, or high concentrations that exceed freshwater bathing thresholds. Our microbial source tracking is telling us something different. And that's what we're kind of struggling with, is we have a little bit of conflicting information. The FIB stuff says there's an issue. There's an issue that happens after a big rain. And we're going to be looking at that closely, and different watersheds have different concerns and impacts and development that may be affecting different watersheds differently. But our microbial source tracking is telling us there's no human waste issue. We don't have, we monitor for human marker, bovine or ruminant, dog, and avian. And we have identified avian Bacteroides at one of our frequently failing beaches, Clam Beach, and we have identified dog bacteroides at a beach that is frequently out, and ruminant, but not human marker has been non-detect so far. So that's really giving us a lot of challenge. And I think when we go to engage the public, we have to be prepared for that conversation. And it can be a sticky one. On one side, you have folks going, high fecal coliforms is indicative of a sewage problem. And on the other side, you can have people saying, why are you all up in my face about sewage because you've got no human marker? So it's just something that we'll have to deal with in our conversations and our strategy for identifying true sources and getting some cleanup. So just wanted to share those challenges. And I guess one other question for the group would be your experience and understanding of Enterococcus and background levels of Enterococcus unassociated with anthropogenic causes. Thanks for that.
So Mario, to just try to answer your question, I think Enterococcus is one of the fecal indicator bacteria that's prone to regrow. So it's possible that it's regrow or some other natural sources instead of human sources.
Yeah, that's kind of what we feel, because we can see exceedances for Enterococcus in watersheds which are marginally developed. I wouldn't call them urban by any standard. Rural residential might be a more appropriate description, and Enterococcus can be exceeding the freshwater bathing limits, but the other FIB will be much lower. Thanks for that.
Looks like Hal has his hand up too.
Yeah, Mario, where are you doing your source tracking?
Our public health lab here in Humboldt County is associated with the Sonoma County Public Health Lab, and the previous lab director, Jeremy Corrigan, was doing some specific studies and has a published paper. I can't remember the name of the publication, but it's regarding MST, and he studied Clam Beach and has some published documents that are peer reviewed, and he was the lab director, so he did a couple studies that had some conclusions about the microbial source tracking, and I can share those at a later time if with someone. If you want to send me your email in the chat or send me an email directly, I'll put my email in the chat, and I can share his publication if that's helpful.
Appreciate it.
Great. Any additional questions, comments, information? All right. So we'll move on to our first presenter. Dr. Matthew Verbelia is an assistant professor of environmental engineering at San Diego State University, where he directs the Safe Water Laboratory and teaches courses on design of water and wastewater treatment systems and microbiological processes of environmental engineering. The Safe Water Lab research aims to better understand the health-related environmental microbiological processes and the fate and transport of microbial pollutants in engineered and natural systems. So Matt, I will pass it off to you to present. I think you have co-hosts, so you can share your presentation.
Sounds good. Thank you for the introduction. So yeah, I'm happy to be able to share with everyone here today a study that we're doing in collaboration with the Waterboard and San Diego Regional Board. And because we're sort of at the beginning stages of the study, and we still have not yet had our first swamp, this is part of a swamp program, we haven't had our first swamp data submittal. Unfortunately, I won't be able to share any preliminary results with you yet. However, I'll talk about the study design and then a little bit about what we're doing for this project. I've also added a couple slides at the end for a previous project that we worked on in the San Diego River, also with the Regional Water Board. So we'll go through background, overall goal, objectives, and project tasks, and our sampling and analysis plan. And also Eric is going to present some of the slides. As mentioned earlier, Eric is a graduate student here at San Diego State. So background, there's, as some folks have mentioned, and probably most people here are aware, there's shortcomings associated with the use of general fecal indicator bacteria for assessing surface water quality. Traditional FIB don't correlate with pathogens, and that's especially true for viruses. This has been documented over and over again in the literature. For our understanding that there's two approaches that have been used for establishing water quality objectives at beaches, and there's the risk assessment approach and the reference system approach. And so, for the risk assessment approach, it relies on assumptions about the ratios of FIB to pathogens. So there are some limitations there in terms of like knowledge of those ratios and how they might change throughout a watershed, how they might be different in different watersheds. And the use of quantitative microbial risk assessment, QMRA, to estimate probabilities of infection and usually limit those probabilities to less than 1 in 10,000, 1 in 1,000, sorry. The reference system approach, on the other hand, recognizes natural sources of bacteria that may cause or contribute to the exceedances of water quality objectives, but that differ from those contributed by fecal matters that usually carry pathogens harmful to humans. And so that's the alternative approach, basically taking a reference beach compared with some potentially impacted beaches and, using those background levels to establish water quality objectives. And so both of these have been described in the scientific literature. Reference system here for our study is defined as a water body with minimal impact from urbanization, industrialization, agriculture, but that has natural conditions that are similar to a potentially impaired water body. And for those of you familiar with Southern California, there's probably very few true reference systems. However, I'll talk a little bit about some of the reference systems that we're using in this study. Overall goal, objectives, and project tasks. So our overall goal for this project is to establish a reference system data set in Southern California with multiple beaches that includes alternative, general, and human-associated indicators of fecal contamination. So the specific objectives, we're measuring the following microbial groups during dry season, wet season, dry conditions, rainy conditions, and we're doing it at reference beaches and impacted beaches. So we're measuring the traditional FIB, E. coli, and Ddpcr. We're also measuring concentrations of viral fecal indicators, somatic Cauliphages. We are using Qpcr and digital PCR methods to quantify HF183, PMMOV, and then we're also looking at some human pathogens like adenovirus and norovirus. If anyone's unfamiliar with pepper mild mottle virus, this is a kind of like HF183, it's associated with human fecal contamination, but it's based on diet. So it's a virus that infects peppers, and peppers that are infected with this virus tend to get ground up into powders and chili powders and sauces, and then they get consumed by humans, and they are very durable in the environment. So they persist much longer than other viruses, and therefore have been proposed as a viral human-associated fecal indicator. So our sampling and analysis plan, looks like this. We designed the study as a three-way analysis of variance, or ANOVA. So the three factors are development impact, and again, that's the impacted versus reference beaches. Sample location, we're collecting samples from ocean and also from estuaries during ebb flows. Season and weather is the third factor, and we're binning that into three categories: summer dry conditions, winter dry conditions, and then winter rainy conditions. We're aiming for a total of 360 samples, and that was initially based on budget, but I'll talk a little bit about how we confirmed confirmed that sample size using a power analysis. So if we just start with like assuming a balanced design for ANOVA, meaning that all the samples are equally allocated into each of the groups, and we used alpha level of 0.05, which is typical for statistical significance. And then we also assumed 80% power. So power being, the probability that when you detect significant differences, that it actually represents a true difference between the groups. And that's the statistical power analysis is the way that you can confirm if your sample size is large enough to detect the difference that you're hoping to be able to detect. So based on this power analysis, we determined that 360 samples divided into these groups should allow us to detect a difference of even less than 0.5 log units between reference and impacted beaches. And we felt fairly happy with that outcome, given the fact that smaller log differences might not be important, for regulators. So that's the sample size determination. Next step was site selection, and through conversations with the regional board, we learned there's a desire to include more than one reference beach and more than one impacted beaches. So that does mean, distributing our samples through several, to several different sites. But we started out by looking at water bodies that were listed as impaired. Given that we were working with the regional board, we were looking at water, basins that were coastal basins in Orange County, as you can see here, and also in San Diego County. So we did a combination of site visits and then also looked at some data about the watersheds in order to narrow the list down to 4 or 5 candidates. And I'll pass it over to Eric to talk about the sites that were initially selected.
So the sites that we initially selected were Aliso Creek, San Juan, San Luis Rey, San Mateo, and San Onofre. The impacted sites, specifically Aliso Creek, San Juan, and San Luis Rey, were chosen due to a desire for the inclusion of a variety of local conditions. For example, San Luis Rey had the highest percent agriculture of all the sites. As for the reference sites, these sites were chosen based on their watershed size and thus how well they could be compared to the impacted sites. For example, San Mateo was chosen because it was similar in size to San Juan. San Onofre was chosen because it was similar in size to Aliso Creek. San Luis Rey, however, did not have a comparable reference counterpart in terms of watershed size. We also chose these sites based on their presence of USGS gauges for flow rates. Here on the top right of the slide is a comparison between each site, showing their respective number of days logged in the seeding database, with Enterococcus levels above the statistical threshold value found in the statewide bacteria objectives for Rec 1 waters out of the total number of days with available data. Here we have some photos of the 5 initially selected sites from when we visited each site for preliminary visits. These are photos of each site's estuary, or the mouth of the stream that can be at times connected to the ocean. One last thing to note before we move on from site selection is that although these 5 sites were initially selected, we are currently only working with 4 sites now, specifically San Juan, San Luis Rey, San Mateo, and San Onofre. This action was taken due to logistical and safety issues encountered when sampling the first couple of times. For example, due to its relatively far distance from our lab, we were struggling to reach holding times for bacterial analysis. In addition, As we can see in picture C, the little sand berm between the ocean and the estuary. Recreational surfers were digging that out in order to create a standing wave, which made it very difficult to sample. In total, at each of these sites, we are planning to collect at least 360 samples. Of these 360 samples, we distributed them as follows in the chart. The non-balanced design was intended to increase the sample size where non-detects were more likely to occur because a larger sample size will result in a higher likelihood of detection. As a result, we chose to collect more samples during dry weather conditions because it is under these conditions where we are more likely to get non-detects. Likewise, we chose to collect more samples from the beaches and less from the estuaries because we expected the microbial concentrations to be lower in the beaches than the estuaries. Another reason why we chose to collect more samples during dry weather conditions was also in part because rain is not the most frequent event in Southern California. For estuaries, our goal was to collect samples at ebb tide or when the estuary is flowing toward the sea in order to gain a more representative freshwater sample. As mentioned earlier, this study will be analyzing traditional FIBs, viral general fecal indicators, human-associated bacterial fecal markers, human-associated viral fecal markers, and enteric virus pathogens. The methods used to analyze them are as follows. For enterococci and E. coli, we'll be using EnterAlert and CallAlert, respectively. These methods are depicted on the top right hand of the slide. In EnterAlert, wells will fluoresce blue when Enterococcus is present, and in CallAlert, wells will fluoresce yellow when E. coli is present. For Cauliphages, we'll be using the single layer agar method, which is depicted on the bottom right of the slide. For the single layer agar method, plaques that form indicate the presence of Cauliphages. And lastly, for the remaining 4 microbial groups, PCR and digital droplet PCR. PCR and digital droplet PCR will be used for the 2 single-stranded RNA viruses in our study, such as pepper model virus and norovirus. Their PCR process will have the extra step of reverse transcription, hence RT-PCR in the chart. It is also important to note that these 3 of these methods that we are using, specifically the ones for Enterococcus, E. coli, and HF1E3, are standardized. However, the rest are not standard methods and are instead based on pre-existing literature. I will now be passing it back off to Dr. Matt Verbelia, who will go over more specifics of PCR and digital droplet PCR.
Okay, we'll talk first just about our sample plan. So we're sending out 3 1-liter bottles for grab samples to be collected from each site pretty much weekly, sometimes every 2 weeks, in order to get the 360 samples over the course of 2 years, which is the timeline for the project. We split them into 4 aliquots. We use 2 of them for IDEX, CallAlert, and EnterAlert. And we use 1 of them for the cauliphage analysis, and then 2 aliquots used for molecular biology. We do separate filtrations for HF183 and viruses. And basically the reason for that is to concentrate viruses from samples. We use the adsorption extraction method. So basically you modify the pH of the sample to bring it down to an acidic pH, which modifies the surface chemistry of virus capsids, allowing them to adhere to the membrane. Otherwise viruses are too small. They would go through the pores of typical membranes that are used. So that modification of the pH is not consistent with the EPA method for HF183. So we do the HF183 filtration separately. So we're planning to hold samples for extracted nucleic acids for at least 3 years. We keep the actual sample only for 2 days. But we will be sharing some of the extracted nucleic acids with SQUIRP. I think they're interested in looking for some Vibrio species and to complement some of the data that we're already collecting. Yeah, I included a slide, just a little background on Cauliphage analysis. I know I've seen in Orange County, you'll have done Cauliphage analysis before, at least in water, wastewater plants, and maybe at beaches as well. I'm not sure, but if you're unfamiliar with this analysis, you grow a lawn of E. coli on a plate, and then you add your sample to the top of that E. coli, lawn. And if there's any coliphages present in the sample, they'll infect the E. coli and leave a plaque. So it's kind of like the membrane filtration for bacteria, where you see colony forming units, only these are sort of the inverse of that. They're plaques in the colonies. So you call them plaque forming units, and each plaque forming unit presumably started with one virus. And so by counting these on a plate, you can backtrack and figure out the concentration of viruses in the initial sample. There's two groups that are specified in the EPA method. The somatic cauliphage group includes DNA genome viruses, so like Phi-X174, which are a little bit more similar to parvoviruses, and they have a single-stranded DNA genome. There are also T3, T7, Lambda, T2, T4. These are all somatic caulipages, double-stranded DNA. They have different shapes from human viruses like adenovirus, hepatitis, but they fall into the same group in terms of their genome. And then the F-specific cauliphages are mostly RNA viruses. MS2 is one example, and these are similar to some other human viruses like poliovirus or rotavirus, etc. So yeah, it gives us a glimpse that's a little bit different from bacteria, right? Because viruses survive, behave differently than bacteria in the environment. And then I didn't want to spend too much time on this, but in case there were questions regarding digital and Qpcr, and I'm really happy to see a colleague from San Diego County here, and I know San Diego County is now implemented digital PCR, which is fantastic. So with Qpcr, we use a standard curve with reference concentrations of the genome that we're looking for. We used Hackman based Qpcr in our lab, so we use probes in addition to 2 primers that bind to the amplicon of the organism that we're looking for. And when those strands get replicated, it breaks off the probe, cleaves it into 2 different parts, which allows the fluorescent signal to be read by the machine. So the more copies that are generated, the higher the fluorescent signal gets. And when it crosses a threshold, we use the cycle number at which it crosses that threshold to determine the concentration in the sample. On the contrary, droplet digital PCR or Ddpcr works more like the IDEX method, where you partition the sample into smaller partitions. Only in this case, you're generating like 10,000 or more droplets, nano-sized droplets. And inside each of these droplets, you may have 1 or 2 or 0 copies of the amplicon that you're looking for, like HF183 or PMMOV. And then the machine reads those droplets one by one and determines, after 40 cycles of PCR, if there's amplification of the genome in the droplet, it'll have a high fluorescence reading, and you can distinguish the positive droplets from the negative droplets based on the amplitude. And so that's how you can back calculate the number of copies of the virus or the bacterial marker in the original sample. The benefits of Ddpcr compared to Qpcr is that you get direct quantification, so you don't have to rely on a standard curve, and it's less susceptible to inhibition from environmental inhibitors. And then you can often get a lower limit of detection. And so we're using Ddpcr strategically probably for samples mostly in the summer when we expect the concentrations to be lower. This is just a glimpse at our team. So we're working with Waterboard Region 9. Chad is the main contact here for Region 9. Helen recently left her post at Region 9, and now she's with a different regional water board, but she's still providing some feedback to the project just based on her interests. And Eric, as you mentioned, he's interning at the regional water board, but he's also a student at SDSU. So we've got a team of 3 students that are all graduate students, and then other students who come in and out of the lab for internships or for learning experiences. plus our lab technicians and lab managers who have helped us, document the QA/QC. And then at the State Water Board, we're working with the SWAMP and QA/QC team for data, in particular, Charles. So yeah, if you've never worked with a university lab before, just a little bit about our team and facilities. So we've got Qpcr, digital PCR capabilities. We've got a small microbiology lab, BSL2 certified in the engineering building at SDSU, where we can do culture-based and molecular biology. And it's been a great opportunity for students who, a lot of them have actually gone on to work for the Waterboard. Several of the graduates from my lab are now working in various units of the State Waterboard or the Regional Waterboard. Just to give you a glimpse of the timeline for our project. So our first data submittal will be in June. And at that point, we would be able to present results, preliminary results from the project. And we've got a few periodic data submittal deadlines, but our final report will come out in early 2024. So yeah, if you're interested in learning about the results, be happy to come back and present once we're kind of further into the project. I don't know if I have a little time left. I wanted to share a little bit about a previous project we did, just because I heard some conversations in the opening about interest in this distinguishing between different sources. Something we're calling like next generation source tracking, because you now have this challenge where you can determine if it's human or if it's dog or if it's gull or if it's bovine. But if it's human and it's human fecal pollution, now the question is, like, where is it coming from? Is it from homeless encampments? Is it from sewer? Is it from septic? Is it from stormwater? So another thing we're working on at SDSU is development of these kind of next generation source tracking methods. And you can download our report from the San Diego River Contamination Study. Our final report was published a couple years ago, and we're in the process. We've already published some of them in peer-reviewed literature, and others are currently in review. But just to give you a glimpse of what we have in this final report, we were mostly focused on trying to distinguish homeless encampments from other sources of fecal contamination. So one of the studies we did was an upstream-downstream study, where we isolated active homeless encampments and collected samples upstream and downstream and compared concentrations of microbial constituents. We also did sequencing on those samples, 16S microbial sequencing. And basically, in short, the result from that was that we did not find any evidence that the encampments were changing the microbial concentrations or relative quantities between the upstream and downstream sites during dry conditions, with the caveat that there may have been like some sporadic instances of contamination that may have happened during one day of sampling, but was not a persistent source of fecal pollution to these three sites. Another experiment we did was isolating encampments that had been abandoned and finding sites with evidence of open defecation. And then we designed a simulated runoff experiment. So we did this on 9 different sites and poured synthetic rainwater over the site to simulate sheet flow, and then captured that runoff and analyzed it in the lab. One of the main takeaways from that is we detected PMMOV at much higher concentrations than HF183. And that's a reflection of the fact that pepper mild mottle virus is more persistent in the environment compared to HF183. And we also detected sucralose at much higher concentrations relative to caffeine. And that's again because sucralose is more persistent in the environment compared to caffeine. So one of the things we're looking at is the utility of using more and less persistent source tracking markers like sucralose and caffeine and HF183 and PMMOV to distinguish newer versus older sources of fecal contamination. And so another paper we're working on right now, which is currently under review at a journal, but it's described in the final report, is distinguishing based on the use of tracers, flow that's more consistent with sheet flow, more consistent with interflow, and consistent with groundwater base flow. And then we collected subsamples during several storm events, every hour or every 2 hours, and tracked the levels of fecal indicator bacteria and other pollutants during these different flow regimes, dominated by sheet flow, dominated by interflow, because we would anticipate homeless encampments to contribute more fecal pollution from sheet flow, whereas leaky sewers might be more relevant when you have interflow happening. So yeah, that's, if anyone's interested in that, I'd be happy to share the journal article when it's published, but we have preliminary findings described in our final report from the study. These are some takeaways that you can find in the executive summary of the final report. Lack of evidence that homeless encampments had a significant influence on microbial pollution during dry weather. However, we did find evidence of microbial contamination of soils at homeless encampments, even after site cleanup. We looked at the persistence of wastewater spiked soils in a lab-controlled experiment, and we found that when flushed with synthetic rainwater, they were still a source of E. coli, enterococci, HF183, and PMMOV after 4 months. However, the levels and the persistence of these different indicators were different, as time progressed. Caffeine and HF183, I talked a little bit about, that these less persistent markers versus the more persistent markers. And then one of the conclusions here was that we felt that the evidence was mostly pointing to untreated wastewater being the main source of San Diego River pollution during storm events. So that's all described in the final report. And I'd be happy to share more details if anyone is interested afterwards. So yeah, thanks for your attention. And if you ever are interested to work with students and work with faculty on a research project, I'd be more than happy to talk about what capabilities and services we could provide. Thank you.
Thank you so much. We're gonna open it up for any questions right now, and we're ahead of schedule. So if you do want to go into further depth, you're more than happy to. But it looks like Luke Ginger has his hand up right now.
Yeah, hi. Sorry, excuse me. Yeah, thank you for that, Matthew and Eric. That was an awesome presentation. I had a question about that wastewater spiked soil that you just mentioned. What did that experiment look like? And did you use sand? I guess when you mentioned that, I was picturing like a stormwater lagoon on like a beach, and is that leaching stuff into the ocean over time? Yeah, could you just maybe elaborate on that experiment a little more?
Yeah, definitely. And first of all, I just want to acknowledge that finding was part of this previous project. I felt bad about not presenting any results to you from our current projects, so I threw in a couple slides of this previous project just to give you some content. But that one was funded by the State Waterboard, but also by the San Diego River Conservancy. And yeah, let me talk a little bit about that persistence study in water flushed from riverbank soils. And I can drop the link to the publication in the chat, but this one was published in a journal just last month, or a couple months ago, sorry. The experimental design was basically like this. We collected soils from the riverbank at a location that was, unlikely to have fecal contamination, and we verified, that the soils were absent of fecal pollution before the experiment. Then we spiked soils in a bunch of these replicate tubes. We spiked it with untreated wastewater collected from a local treatment plant here in San Diego. So we did that, to several replicates of the tubes. We had two kind of dimensions of the experiment. The first one was a consecutive flushing experiment. And so after spiking with sewage, we waited overnight just for it to kind of dry out a little bit. And this was all performed in the laboratory, no sunlight exposure. And then we flushed it with synthetic rainwater once. Then we had some where we flushed it twice, 3 times, 4 times, 5 times, up to 20 consecutive flushes of synthetic rainwater. and then we measured the concentrations with each consecutive flush. The time delay flushing experiment was with a single flush of synthetic rainwater. However, we waited different number of days after spiking with sewage. So 14 days all the way up to 121 days. And so the results here, and we measured for TDN and DOC just to get an idea of organic carbon in the flushed water. But for the consecutive flushing, you focus on the bottom left panel. Basically, we found that E. coli, Enterococci, HF183, PMMOV, they were more or less being released at similar rates if we normalized, to this different starting concentrations in the sewage. After 20 flushes, the concentrations in the flush water was much lower, and they sort of declined at a similar rate. However, in the time delay experiment, the one where we spiked and then we waited a different number of days before flushing, we saw that, as expected, PMMOV signal was still very high even after 121 days. HF183 and Enterococcus was, that level was kind of in the middle. E. coli had the biggest decline over time. And just to I guess as a qualification here, E. coli and Enterococci were measured using culture-based methods, IDEX, whereas PMMOV and HF183 were measured using PCR-based methods. So, the PCR doesn't necessarily distinguish if it's alive or dead. So there's a difference in methodology, but that was an interesting finding from this study. And we were trying to simulate what might happen if there's a sewer overflow and it sits around in the soil for a while, and then later there's a rain event with interflow that, potentially could wash that contamination into the river.
Thank you. That's awesome. And yeah, if you could drop that that link in the chat, I would appreciate it. That's a really cool experiment you did, and you're kind of getting at the first flush event a little bit there too with an in-lab experiment.
Thanks. Yeah. Put the link to that paper right there in the chat. And I can also put the link to our final report for anyone who wasn't able to get the QR code. But again, and just to reiterate that study was a separate project from the one that we were invited here to present. So we just don't have data from our current project, but hopefully next time we come through, we can share some of the results there. The time for, I see some other raised hands. Yeah.
Eric and Brian, if you could hold off, I think Gian was first.
Oh, thank you. Quick question. I think a lot of the findings are very interesting about homeless and probably other sources, but in the end, you said the bacteria or the pathogens are mostly coming from the untreated wastewater. I know that there's County of San Diego and other folks are doing source tracking and mitigation for quite some time. I don't know if there are any findings where this untreated wastewater is from, and if you can elaborate that, it'll be very helpful. I'm just curious. I know there are probably RV dumping or some septic tanks, but do you have any further information on that one?
We didn't isolate specific locations or sources of untreated wastewater in this study. Like I mentioned, we used various source tracking markers, both chemical and microbial, and also some tracers to try to distinguish sheet flow from interflow from base flow. And the evidence was mostly pointing to, not that it was the only source, but most likely the dominant source. And that is a finding that's described in the report in more detail. We're currently involved in follow-up studies, and not just my lab, but also Professor Natalie Mladenov at San Diego State. She's got some ongoing projects where they're looking further into, sources of fecal pollution, again, to the San Diego River. And then, of course, we're using the source tracking markers and some pathogens at the beaches in the study that I described, Eric and I described in the first part of the presentation. So I'm not sure if that answers your question, but yeah, we didn't specifically isolate, which sewers, although I'll note that the San Diego River, the main sewer trunk sort of runs all along the river. And so one possible source is, leaky sewer systems or junctions of sewer systems. And I know SQUIRP, and I see some folks, I think, here who have joined from SQUIRP. I know SQUIRP has also been looking into that.
Thank you.
Eric, if you'd like to ask your question now.
Yeah, thanks. I'm interested in the relative persistence of the various human markers, and I'm wondering if you think you can develop that into a clock, so to speak, of aging, to age the waste and relate that to pathogens, so that you can get a sense by looking at maybe 2 different markers, how old the waste is and how likely you would have a particular pathogen load.
Yeah, definitely. So we know that there's wide variety in persistence from different pathogens, from protozoan, bacterial pathogens, virus pathogens. There's lots of evidence in the literature that the persistences are very variable between different pathogen groups. And the same is true for indicators. If you talk about, traditional fecal indicator bacteria compared with Cauliphage, for example, like we mentioned that we're studying, to PMMOV, which is incredibly persistent, and then Crassphage is another virus that people are using now, human associated. And then the bacterial markers, HF183, perhaps a little bit less persistent compared to PMMOV and Crassphage. So yeah, that's what we're trying to focus on now with our current research, is again what I kind of call like these next generation source tracking methods, where we know it's human fecal pollution because of the source tracking markers, but can we tell something about the age of the fecal pollution, or can we tell a little bit about where it's coming from, if it's coming from subsurface sources or if it's coming from surface runoff? And I think that we might have some hypotheses here that could be interesting to look further into. And we're working on NSF proposals and try to get some funding to follow up with this EPA proposals. So we're hoping to continue to investigate this topic in our group here at San Diego State.
Awesome. And then Brian.
Yes, thanks again, Matthew, for the great talk. I was just curious about the Cauliphage analyses because here with the city of San Diego, we recently were a part of a SQUIRP study with 1642. And I was just kind of curious as to your lab's experience on just getting accustomed to it, because, from our experiences, it a little time consuming and can be a little challenging to learn. So I was just kind of curious as to your guys' experience to that. And also, how did you differentiate between, or when you did 1602 versus 1642?
Yeah, I'll mention, so we actually haven't started analyzing samples with 1642 yet. We plan to do that now that we're entering the summer, because we've been using 1602 up until now, and actually we've been getting detection. So there hasn't been a need to move to 1642 yet. And I guess for everyone, if anyone's unfamiliar with the 2 EPA methods, 1642 involves concentrating a much larger volume sample using ultrafiltration membranes, whereas 1602 involves directly analyzing 100 milliliters of the sample, which is like kind of the maximum that you could fit in Petri dishes. So speaking to your first part of your comment and question, is like the training on that method. There's a big learning curve, And I'd say I start with the students sometimes when they're undergrads, having them learn the method. And then by the time, if I can recruit them into the master's program, by the time they start the master's program, they at least they're proficient with the method. And then, that's the way we've done it so far. With 1642, there's definitely an increased cost just with the materials, the amount of time it takes to filter the samples. Now while we haven't done 1642 for coliphages, we have used ultrafiltration membranes in my lab for concentration of microorganisms and viruses from large volume potable water samples for a different project. So we've done the up to 100 liters of filtering advanced treated water using ultrafiltration membranes. And it's time consuming, it's material intensive, the membranes are expensive, and it definitely increases the cost. But I think if you're comparing that with the other side, which is like getting a bunch of non-detects because your concentrations are lower than 1 per 100 milliliters, then it might be worth it in some cases. And especially I think if you can combine, maybe some of them get analyzed for cauliphage, but you extract nucleic acids from another portion of the subsample, and you might be able to get good value out of it, especially during periods where you anticipate the concentrations to be low.
Thank you.
I'm not seeing any more hands. Are there any more last minute questions? Okay. Well, thank you so much, Matt and Eric, for presenting today and taking the time this morning to join us. It was, from what it looks like, a very stimulating conversation and really interesting. So thank you so much.
Thank you. Appreciate the opportunity.
And we are running about 15 minutes early, but I'm going to hand it over to our next presenter, Kat Walsh, the Ocean Unit Supervisor, and she's going to be giving a presentation on Coastal Beach Monitoring Program's data quality goals.
Thanks, Michelle. Yeah, I'm just going to share my screen really quick. Is it in presentation mode? There we go. All right, yes, so I'm going to present on our data quality goals for the Coastal Beach Monitoring Program. Again, I'm Kat Walsh, I supervise the Ocean Standards Unit in the Division of Water Quality. All right, let's go. So just a bit of history, I'm sure you all are well aware of this, but the beach program was established through the passing of AB 411 in 1997. And then in 2012, there was a change in how the program is administered through the passing of Senate Bill 482. And so what that did was kind of split the responsibilities between California Department of Public Health and the State Water Board. So the State Water Board assumed the responsibility for directing, monitoring, managing the statewide database, and providing funding to the 16 local environmental health agencies that implement the monitoring program. And then CDPH retained the responsibility for establishing and amending minimum standards for sanitation at public beaches. So many of the local agencies are aware they've been working on quality assurance for the past several years in developing their quality assurance project plans, but we've also been evaluating and taking steps to improve quality assurance for the program pursuant to 40 CFR 31.45, which includes documenting a systematic process to ensure that the quality of data generated is known and is appropriate for decision-making purposes, as well as minimizing the loss of data due to out of control conditions and malfunction. So we're this is a part of our receiving federal funding from the US EPA. It's a requirement that we have a systematic process for documenting quality assurance and assuring that the data is of known and appropriate quality. So in doing this work, we provided a report to the US EPA in January of this year, in which we identified the 4 goals for improving data quality assurance for the program, and our approach for developing a quality assurance program plan for the beach monitoring program. And we also established a process for reviewing our quality assurance efforts, as well as looking at opportunities to improve the beach program overall. So I just want to share a quick reminder of the tiered quality assurance structure at the State Water Board. So again, here's the quality assurance project plans that many of the local agencies who are participating today are familiar with. You have developed a draft quality assurance project plan. And nested above that is the quality assurance program plan, which is the document that we're going to be working on over the next few years. And then above that is the quality management plan, or QMP. That's for the State Water Board entirely. So it's kind of nested down, and the SOPs, the standard operating procedures, are at the bottom, and they're supposed to be conformance upwards. So quality assurance project plans are supposed to be in conformance with the program plan and the QMP, and also similarly, the quality assurance program plan is to be in conformance with the State Water Board's QMP. So our 4 data quality goals are establishing data acceptance criteria, and what that is identifying program-wide acceptance criteria that data will need to meet in order to minimize the possibility of making erroneous conclusions. And to do this, we're going to identify and document data quality objectives, data quality indicators, and measurement quality objectives for the program. This could include establishing acceptable levels of uncertainty related to precision, accuracy, and representativeness, as well as identifying criteria to minimize or control sampling and measurement of error. And then another goal is to establish control measures, quality control measures. And that is to, in doing that, we'll be establishing standardized quality control activities to verify and validate the quality of data generated. And that will include identifying the type and frequency of quality control measures to be implemented for field sampling and laboratory analyses, such as replicate sampling, field duplicates, and sample holding times. And then another goal is to establish procedures for data verification and data validation. And our goal for this is to develop a systematic process to ascertain the data's completeness, correctness, and consistency with methods and acceptance criteria for the program. This will include identifying sampling and analytical information that should be reviewed during the data verification step, as well as establishing procedures for evaluating analytical data to verify it meets the acceptance criteria, and labeling data that does not. And that might also include corrective action procedures for the data that does not meet the acceptance criteria. And then our 4th goal is to evaluate the data management strategy. And that considers the data life cycle. And our goal is to evaluate measures to further monitor, control, and store data that is generated for the program. And this might include updating our Beach Watch database to allow the local agencies to submit data quality quality control data with the data that is submitted already to Beachwatch, and to identify the data or flag the data that doesn't meet the acceptance criteria. So that's a little bit about our 4 key goal areas. And then tied in with this, because this program's been in place and monitoring's been conducted since 1997, we recognize that it's challenging to come in now at this time and restructure the data quality control measures and quality assurance measures program-wide. And so we've come up with a phased approach for developing and implementing the data quality assurance program plan for the BEACH program. And so phase 1 is the phase that we're currently working on, and our goal is to complete this by December of this year. And that's really where we're focused on documenting existing quality assurance, quality control, and data management procedures that are already exist program-wide, and then also identify and establish initial acceptance criteria for the BEACH program that would apply program-wide. And then there's phase 2, which would proceed after that is approved, is to refine our acceptance criteria if needed and establish quality control procedures and initial data verification and validation procedures for the program. We are hoping to complete that by December of 2023. And then phase 3 is to refine the quality control activities and the data verification and validation procedures if needed, and evaluate and implement a data management strategy. And for that, we are hoping to complete phase 3 by December of 2025. 2024, I think. My apologies. So we're phasing this each year. We're going to try and complete these steps, but we do recognize that there may be some challenges or some delays in implementing this. So we'll be evaluating every year our success so far and whether or not we need to focus on a different component of the data quality assurance program plan. So there's some additional considerations that we have, and this is kind of the purpose for our latter part of this meeting, the closed session for the local agencies, as well as some other opportunities that we'll be hosting through the course of the year, is that we want to provide opportunities for data users, data producers, and decision makers to be involved in each phase of developing the quality assurance program plan. And this is to ensure that their needs are met and are defined and addressed in the program plan, and also to collaborate on improvements for the BEACH program and try to build consensus and understand the challenges around implementing our quality assurance, quality control, and data management activities. And then we also have a second consideration that we're focused on, is to annually review the program needs and the lessons learned. This is not just for the quality assurance program plan. It's program wide. So this would include evaluating the effectiveness of existing analytical methods, needs around that, considering opportunities to further expand the use of rapid detection methods such as Ddpcr. It's also tied in with some of the recent conversations that we've been having around funding for the program. You may have provided some responses to Michelle on the funding needs for your specific local agency recently. We're really trying to figure out how we can continue to improve and move this program forward. So it's a brief update on our goals and what we want to do. And we'll go much more in detail for our development of the program plan in our next session. Michelle and our quality assurance manager at the State Water Board are going to host an open discussion on what we can do around a systematic program-wide approach for quality assurance. But here's the contacts for everybody who's working on this right now. Myself and Michelle are probably going to be the leads for phase 1, but Amanda will be assisting. And then for phase 2 on, Amanda's going to take lead for developing our program plan. With that, I'll take questions.
Thanks, Kat. There is one question in the chat from Keith Allen, and he's asking if there's any updates on the status of merging and or bridging Beach Watch and CDIN Waterboard water quality data programs together.
Yeah, there aren't any updates. From my understanding, we are going to continue to use Beach Watch. Beach Watch has been interconnected with CDIN, so any data that is entered into Beach Watch has been transferred over to CDIN, with the exception of the postings that are submitted. So all water quality data, though, is transferred to CDIN. It's just those, I believe they're called flat files, that aren't accepted into CDIN. Keith, I see you have hand raised.
Okay, it's probably easier if I just ask verbally than typing. So we currently have to enter into both systems separately. There's nothing that's, Beachwatch is a heck of a lot easier to use than CDIN. CDIN's very, it's very technical and it's very finicky. So I know back in like 2018, 19, somewhere around there at a SQUIRT meeting, it was discussed that we would hopefully in the future, we would only have to enter into one and then it would carry over. But that doesn't seem like it's happened yet. And it's pretty labor intensive for our clerical team to have to do that entry. So I was just curious if there was any updates on it to help alleviate our sorrows over here in Long Beach.
Yeah, it may have been that the conversation was around that interconnection between Beach Watch and CDIN, and there might have been like a side conversation about just using CDIN alone, but that was not a part of our project back in 2018, but I have added it to the list of things that we should be considering for the beach program.
Okay, thanks. Yeah, I think that was what they kind of kicked around the can, was that they were going to just use CDIN, but I understand it's two different sections of the water board that are looking at these sets of data. So great, thank you.
Actually, Kat, this is Andrew Hamilton, if I may, Keith. Just a question about what you're asking. Is this data for the beach program, or is it data for a swamp program that you are using to satisfy both program requirements?
So it's both. So basically we do our beach sampling for all our sites on Mondays, and so that's entered into both because Public Works Department in Long Beach, they've actually contracted with us to use our sampling data, which we have to enter into CDIN. And then we also do 2 additional days, Tuesdays and Wednesdays, for part of the beach closest to the LA River. And we also have to enter that into CDIN as well. So it's satisfying both agencies' requirements.
Okay, great. That actually will probably lead into our discussion moving forward in next session about the ways to verify data. And it really comes down to Swamp has a more robust quality assurance program plan. and requirements for the QA. That's why the CDIN requirements to enter all that data is very much, it's more, like you say, it's more technical, but there's more information that's requested. And that's because of the quality assurance program plan on SWAMP. What they require to verify the data is of known quality. And that will be a discussion that we'll probably have a next session about how we can either unite them so that there's consistency across those programs, or find a way that you can then have that Beach Watch be a place where you can enter for both. So I think that's a discussion that we can have later on.
Cool. Thank you.
Thanks, Andrew. Any other questions on our data quality goals? Well, I look forward to discussing this more in our next session. Michelle, did we want to take a break before that?
Yeah, so we're gonna do a 15-minute break. The next session is our closed section to discuss the beach program quality assurance with the local agencies. So if any of them are on the live stream right now and you'd like to join in on the discussion after the break, please send an email to the beach program email, dwq-beach-monitoring@waterboards.ca.gov, and we'll provide the Zoom meeting information for you to attend. Other than that, let's meet back here at 10:45.